Hello and welcome back. Today is all about the origin of cells. Where did they come from? How do we know this? What are some working theories on the inner workings of cells? So let's dive right in with a recap of the cell theory. Now we have talked about this before, but I'm just going to put it down again because this is the current understanding of how cells work, right? All organisms are composed of one or more cells. Cells are the smallest units of life, and all cells come from pre-existing cells. There was a previous hypothesis, I'll say, that spontaneous generation was what caused cells to just appear, what caused microorganisms to just appear, and Louis Pasteur disproved this by doing an experiment on broth, and you can see that on the right-hand side. So he had a flask that had a curved neck, and so he would boil the broth, and the steam from the boiled broth would come up and end up sitting right here, so water would condense again and form kind of like a barrier right at the bottom of this neck. And so if the neck was still attached and the water was there, the broth would remain free of microorganisms, but he also did the same thing and then broke the neck off. And when he broke the neck off, there was no longer a water barrier preventing things from getting inside of the broth. And when that happened, the microorganisms that were in the air were able to land in the broth and grow. So this experiment disproved the thought of spontaneous generation and proved that all cells have to come from a pre-existing cell. They have to come from somewhere. Now, I said that was the cell theory, but as we all know, there are always exceptions to the rule. And here are some examples of exceptions to the cell theory. So a couple specific examples are striated muscle fibers, which is like the muscles in your body. They're large with many nuclei inside, which is not very typical of cells, so it doesn't really fit in our current understanding of how cells work. Aseptate fungal hyphae, so the hyphae that makes up fungi, they're sometimes, not always, but sometimes not divided up into individual cells, but like the definition of hyphae are single cells, like really thin. They look like long pieces of spaghetti, basically, just like tons of little thin cells lined up one right after another. But sometimes they're not divided up one right after another into individual cells, so you have multiple organelles that typically only have maybe like one in a cell. You have multiple organelles in a hyphae cell. Giant algae is another one. So sometimes these algae can be as large as 100 micrometers and still considered a single cell. And as we know, the surface area to volume ratio is like mega important for a cell to continue working at like its optimal capacity. So giant algae is like, well, why do you exist if you're not really working as well as you could be? And that kind of goes with number 4, where very large cells with continuous cytoplasm are not compartmentalized. So giant algae is like a specific example, but there's other examples that we see of very large cells. Viruses as a whole, they fit like many of the checkboxes for cells, but they can't reproduce on their own. So that's like a big no. So they're not actually considered alive, but they can do a lot of things that cells can do. And then number 6 is like the million-dollar question, right? Where did the first cells come from? I mean, if all cells come from pre-existing cells, there had to be a first cell. So they must have arisen from non-living material. And that is like, well, okay, so they can spontaneously appear? Sort of. We'll talk about that in the next slide. And then finally, as our final exception, some tissues just have large amounts of extracellular material. So in bones, right, there are bone cells. There are mineral deposits in bone, like, okay. We know why we need them, right, because bones are needed for strength. You have calcium deposits in bone for strength, but You don't see calcium deposits in any other cells. Xylem in trees, xylem is used to move water up and down plants, but xylem are dead cells. So why are we keeping dead cells in, in an organism? And then tendons that connect muscles to other muscles, that they're like tough cells that don't really replicate. They are very slow in healing. So again, these are like extracellular material. Why are they here? We'll talk about this more, but these are really good exceptions to know when you have to discuss the cell theory and where we are in our current knowledge of how the cell theory works. So let's talk about the 4 processes for spontaneous origin of life. This goes back to the previous slide where did the first cell come from? So this is sort of the working theory currently on what happened. So number 1, production of organic carbon compounds such as sugars and amino acids. This was replicated in an experiment by Miller and Urey. So they recreated the conditions of a prebiotic Earth, so before life was on Earth, in a closed system to see if you could produce organic compounds. Organic meaning that it has carbon and hydrogen in it. So water was boiled to form vapor, and then that vapor was mixed with methane, ammonia, and hydrogen, and then it was exposed to an electrical discharge to stimulate lightning. So all of those items, water vapor, methane, ammonia, and hydrogen, was then basically struck by lightning to see what would happen. And after about a week, simple amino acids and oily hydrocarbons were found within this closed system, which means that this was maybe a freak accident, because lightning doesn't always strike, right? And the fact that it did hit this perfectly, there was creation of macromolecules. And macromolecules can be built to create living things. So this was like the beginning. So we've now made our macromolecules. The second step is the assembly of those molecules into polymers. So we went from monomers to polymers. So hot sand, clay, lava, they've been shown to produce polymers from dilute monomer solutions. So just having like a very limited amount of amino acids or oily hydrocarbons, when then exposed to hot sand, clay, and lava, they could produce polymers. So now we're getting bigger molecules, right? Like bigger sets of proteins and lipids and carbohydrates and everything. So then we package those molecules into membranes. So phospholipids are amphithetic molecules that would have naturally formed a bilayer, right? We know this because the cell membrane is made up of phospholipids, right? So phospholipids naturally want to form a bilayer. They naturally want to form a vesicle. So vesicles could form because of these phospholipids, which then allows for internal chemical reactions to occur separate from the environment, right? What's happening inside your cells is not happening like in my classroom when nobody's here, right? There are different reactions happening in my cells. Really, there's different reactions happening within my heart cells versus my skin cells, right? So from there, then we have the development of a mechanism for inheritance, which we think the first genetic material was RNA because it's self-replicating and it can act as a catalyst. So it can replicate itself, it can be passed on without the help of anything else, like a ribosome or anything like that, and it can act as a catalyst to jumpstart chemical reactions. So from this like crazy idea of, well, where did the first cell come from? Well, like this. This is how they think the first cell became the first cell. And now let's talk about our last piece of information, the endosymbiotic theory. The endosymbiotic theory, I know if you break this down, endo means inside, symbiotic is like a relationship between two organisms. So a relationship inside of something? Yeah, you betcha, that's exactly what that means. So the endosymbiotic theory is the idea that the mitochondria and the chloroplast were separate organisms, like at the beginning of time, like millions of years ago. They were a bacterial cell that was then like kind of taken in via endocytosis. It was like brought into a eukaryotic cell, brought into another cell, and then it kind of morphed, evolved into what we now know as the mitochondria or the chloroplast. So this idea was that it started as a bacterium, it kind of came into this other cell and then morphed, and now its like main job is energy production. So this endocytosis of these potential mitochondria, potential chloroplasts, may have actually led to eukaryotic cells. So we have these organelles, and now we're adding these other organelles like mitochondria and chloroplasts, and from there now we're building these really complex cells that can do a lot on their own, right? Creating energy on their own is incredible. So this is the thought process of how eukaryotic cells came to be, because prokaryotic cells are older, much simpler, and have like less they don't have barely any organelles, only ribosomes and a cell membrane. So I said all this, it sounds pretty good, right? You believed me right off the bat. Well, you ought to have evidence for anything that you're going to believe. And so here is our evidence for endosymbiotic theory. This is the last thing we're going to talk about. 4 big pieces of evidence. Number 1, mitochondria and chloroplasts have their own DNA. That means that their DNA is different than the rest of my cell's DNA. My DNA in my mitochondria is the same as my mother's DNA. But that's not mine, right? Because I am not a clone of my mom. Additionally, their DNA is naked and circular like prokaryotes. Whereas, as you know, the DNA in our nucleus is enclosed within a nucleus and linear. Mitochondria and chloroplasts have ribosomes that are similar to prokaryotes. They have 70S ribosomes rather than 80S like the rest of a eukaryotic cell. Mitochondria and chloroplasts have a double membrane. The inner membrane has proteins similar to prokaryotes, again, which gives good evidence to the fact that they were a bacterial type cell prior to being engulfed in a eukaryote. And then finally, they're about the same size as a bacteria cell, and they're susceptible to the antibiotic chloramphenicol, which if you are susceptible to an antibiotic, that means that you're kind of susceptible as a bacterial cell. That's what it's used for, is to kill bacteria. So it again shows that it has bacterial origins. So all this evidence shows that at one point in time, mitochondria and chloroplasts were found outside of a eukaryotic cell, and they were brought in via endocytosis, evolved slightly to fit into the eukaryotic cell, and now are used exclusively for a eukaryotic cell, really, to make energy, whether for animal or a plant. Okay, that's it for today. Great job. I'll see you in class.